Greetings and welcome to EHA Unplugged, the official podcast channel of the European Hematology Association, EHA.
So, hello, I am Isabel Olivera, a medical writer for the European Hematology Association. Welcome to this podcast on the process of CAR-T therapy. I have the pleasure to have here with me today Professor Julio Delgado. Welcome, Professor Delgado.
Yes, I'm Julio Delgado. I'm a hematologist. I work at Hospital Clinic de Barcelona.
So we are going to discuss today what the process of CAR-T therapy involves and what are the critical steps that hematologists have to be aware of. Can you first briefly describe what the process is, just the main steps of CAR-T therapy?
So the first step is to actually identify the patient, obviously. And this could be a problem because not all institutions have access to CAR-T cells, so that means some patients are referred from other institutions. And not all hematologists are aware of the indications that are eligible for commercial CAR-T therapy, etc. So one thing that is important is to discuss this, to have a good network of centers that are referring to you so that patients are referred to you if you're a CAR-T cell treating center as fast as possible, because time is of essence. And we're talking about relapse refractory diseases. It's important to set up all the needed permits and agreements, because there's an experts committee that needs to approve that treatment, particularly in Spain. Other countries may be different, but in Spain you have to go through a series of approvals, and that needs to be done as fast as possible. Once this is done, then you just contact the manufacturer, you set up a slot, a production slot, and then you need an apheresis. So again, it's crucial to have a close contact with the apheresis unit of your institution so you can adapt the apheresis to the production slot that is given by you by the manufacturer. So once this is all set up, then the cells are shipped for manufacturing. While you wait, of course, you have to keep the patient fit, not just alive, but fit. And sometimes the disease is so refractory, you have to give them a bit of chemotherapy, a bit of treatment to keep the disease under control while the cells are being manufactured. When the cells come back, then you need a place where to receive the cells. In some institutions it's a pharmacy, in some institutions is the blood bank where products are stored. Some products come as fresh products, some products come frozen. So you need a bit of understanding of where do you need to store them. And then just decide, admit the patient to the hospital, administer chemotherapy, and then you administer the cells, and that is a moment where another critical part comes to play. The first week after the cells are infused, which is when the most important complications may occur. These are immune-related complications, cytokine release syndrome, neurotoxicity. So the whole team needs to be aware of those, how to treat them, how to prevent them, how to manage them. A good network of physicians in your institution, including neurologists, ICU specialists, etc. After 1 week, normally the patients are okay to go home. And of course you need a clinic where you can visit them as outpatients, check the blood counts, check the immunoglobulin levels to see whether the patients need support. So I would say critical steps are, importantly, the network, so the patients are seen and referred for CAR-T cell therapy early. Then apheresis unit, close collaboration with the apheresis unit, and then good understanding of the early complications immediately after CAR-T cell infusion.
How do you ensure the quality and the viability of the collected cells. And
yes, this is actually quite straightforward. It's not that difficult because apheresis units have been there for quite many years now. So we hematologists, we've been doing apheresis to collect other cells, not just T cells as we do for CAR T cells, but stem cells, platelets, plasma. Physicians' experience in the art of collecting cells or substances from the blood have been there for many years, and they have a very good training on quality, on the tests that you have to do to the patient before collection, the serologies you have to do, how you check on the blood before collection and after, so you can make an estimation of how many cells you've collected, and then blood cultures, all that, those sort of tests that you have to do to ensure that your product has no contamination, etc. And these clinicians, some usually hematologists or blood bank specialists, have a very thorough training, and they've had it for many years now. So I would say that they are pretty good at their job.
And these cells will go either to a private manufacturer industry or an academic center that will transfect the vector that will express the CAR construct. What are the key steps there?
So of course what I've described is the usual situation, which is a commercial CAR T cell product. Of course there are researchers, and I am one of them, who actually manufacture their own cells. And that brings another set of skills that is different to what I've just explained. And this is not something you can do on your own. So I am a hematologist. So the first thing I need to say here is you need collaboration. And not collaboration with one or two guys. I mean, I need big collaboration. So first of all, you need to manufacture the vectors, the viruses that you're going to use to transfect the cells. And you need a lot of expertise to be able to manufacture these vectors in good manufacturing practice conditions. And you will normally need a dedicated team to just do that, to prepare the vectors. Then you need a separate team to actually manufacture the cells with the vectors. So to culture the cells with the vectors and cytokines and other stimulants. And normally you would use a bioreactor. There's a number of commercial bioreactors that you could use. They provide you with everything, all the consumables you need, all the cytokines, all that is provided to you. But you need to learn how to mix them together and do it in the right way. So this is normally done by immunologists, because they are the experts on T cells, on immune cells, and how they behave and how you can grow them properly. Importantly, in both lentiviral manufacturing and cell manufacturing, you need to set up quality control tests. So again, another important issue. You need to understand regulation. You need to discuss everything you do with regulators, so you do them in a right way, so you can then administer the product to the patient. And on top of all that I've mentioned, you need to prepare all the documents that are associated with this, so you can present them to the national competent authority, or even to the European Medicines Agency or the FDA if you're in the States, and have your product approved, your clinical trial approved.
For each patient?
Not for each patient, for each product. But that is a lot, and documents is quite a lot of paperwork. And again, this is not something you would normally do if you're a hematologist. So in my institution, this is done by the Department of Clinical Pharmacology. So they are the ones with expertise in regulation, with expertise in document preparation. And also pharmacovigilance, because once you've decided that you're going to manufacture your own products, you are legally responsible for that product. And that means you have to collect all adverse events, you have to compile them, prepare reports, and update the national competent authority regularly.
So now you have prepared the cells. They have to go back to the hospital or the service that will infuse them in the patient.
Of course, depends if you're making them yourself, then they are there already. Of course, patients need to be admitted to a hospital, and then you give the chemotherapy. When you're shipping the cells to the manufacturer, sometimes this is even across the ocean. When we started, the manufacturing facilities for Novartis and Gilead were all in the United States. So it was a bit more complicated because of that, but big pharma companies actually learned a lot over the years, and they can now, these processes have been optimized.
So you mentioned that you have to prepare the patient. There is the bridging therapy, so waiting for the cells to be produced, lymphodepletion regimen.
Correct.
Before infusing the cells. So this lymphodepletion regimen is done quite often, most often with chemotherapy.
Yeah, it is always done with chemotherapy, and the doses and the drugs that you use are basically what the manufacturer tells you to use.
You don't have a say on the regimen, it's already...
No, because it's an approved product and you have to follow the SMPC of the product, but that's not something that is particular of CAR T cells. That happens with any drug that you use. You have to follow the SMPC. You just can't do whatever you think or change the days or... No, it's actually compulsory and improves the outcome of CAR T cell therapy. Bridging therapy, on the other hand, is not compulsory. If you can avoid it, it's better to avoid it, because adding more chemotherapy, more therapy in between could be a cause for extra toxicity, could delay the CAR T cell administration. For instance, if you cause neutropenia, you can't just carry on. You have to wait until this neutropenia is recovered, or the same with liver toxicity. So sometimes it's a problem, and if you can avoid it, you better avoid it. But in some patients, you must do something, because they can't just wait for a month or 3 weeks until the cells are back. So yeah.
So then you infuse the patient, and you mentioned earlier you have to keep really a close watch
The first
for toxicities,
week.
so
Yeah.
for the immediate toxicities.
Yes.
So this happens obviously as an patient is in,
Yes. Is in the hospital
in the
normally.
hospital.
Yeah, and it's better that way, because these immune-mediated toxicities, they can be quite severe and quite become severe within hours. So it would be actually quite difficult to do it in the outpatient setting. And so because we know the toxicity is closely related to the tumor burden. The higher the tumor burden, the bigger the immune reaction. So if the patient comes to CAR T therapy with lots of tumor cells in the body, then you know this patient is going to have immune-mediated toxicity on day 1, day 2. We administer CAR T cells sometimes to patients that have minimal disease. Sometimes because we've already given them CAR T cells and we give them like a boost dose. In those situations when we know, we have confirmed that there's minimal disease, then we've already administered them in the outpatient setting. So we administer them in the hospital. That the patient has to come to the hospital to get the cells, but then the patient goes home. But this is very specific and very specific situations, and always with our own products. This is something we can do because we own the product and we get to decide how we administer the product because it's our product. But for others' products, well, you have to follow their guidelines.
There has been a lot of discussion because obviously these patients are progressing or have at least the risk of progressing during this time. And everything is done to try to shorten as much as possible this process. How long does it take now?
Yes, the culture time, so the time cells are in the machine, is not that long. It's between 7 and 10 days. But you need quality control tests before you can infuse the cells. And then, for logistical reasons, you may have the product but not a bed available in your ward. So, with our product, the medium vein-to-vein time, vein-to-vein is from the moment the cells are collected to the moment the cells are infused back, is 1 month. And as I said, it has to do with a myriad of things, not only the time you have to manufacture the cells. It has to do with the patient, with finding a bed, with the lymphodepletion chemotherapy, and with QC.
What parts of this process can
the
be
culture,
automated?
the culture. I mean, there are artificial intelligence protocols that can optimize the culture so that the cell production is better and faster, so you can shorten the time. There are clinical trials with one-day culture. But one-day culture does not mean, again, that you extract the cells to the patient and then you infuse the cells 1 day later. Because again, everything else needs to be accounted for. We have participated in a clinical trial aiming at optimizing everything. And in this trial, the vein-to-vein time is 7 days, which is possible, but it's risky also. And it's risky because in order to have a vein-to-vein time of 7 days, you have to start lymphodepletion at a point where you don't know if you're going to have cells or not. You put the cells into the machine, then you start lymphodepletion chemotherapy. But you don't know if you're going to get the desired amount of cells, if the cells are going to be okay or not, if there's going to be contamination. You don't know. And then the QC for this product in this trial is done using optimized technology, so that it's done in basically 24 to 48 hours. So all this can be accomplished. But it puts a lot of pressure also on the physician, because you know the patient is already receiving chemotherapy. So I'm not sure if this 7-day vein-to-vein time is worth it in a way. But it puts a lot of stress on the personnel, and well, it's tricky, but it can be done. And of course, the cells need to be manufactured on site for that. For a 7-day vein-to-vein time, it would be impossible to do it in a centralized manufacturing facility in a different country.
So I imagine there is a percentage of patients that cannot receive this therapy because they cannot wait. That you know they will not.
Yes.
And would it be an aim to shorten the time for this specific patient group, and then for the others allow this month more or less?
Yeah, the problem here is that you don't really know when you select the patient. I mean, you can guess how the disease is going to behave in the following month, but you don't have confirmation for that. What others are proposing is the use of allogeneic T cells. So a way of circumventing that is just not using the patient's own T cells, but using a donor's T cells. But that is a possibility, because with allogeneic T cells you could have an off-the-shelf product. You could have the cells ready for use, and then you just need them. You get them from the fridge and you infuse them to the patient. The data that we have with allogeneic products so far is not as convincing as the data we have for autologous CAR T cell products. They persist less. And again, it's also a question of numbers. Autologous CAR T cell products, more than 34,000 products have been administered over the last decade. For allogeneic products, the number is much smaller, so we have much less experience. But that could be another solution, not using the patient's own T cells. That would also make products cheaper, because when you don't use the patient's own T cells, then you can bulk, you can prepare cells for many patients in one go, and that reduces costs, of course.
So this process is very expensive. How do you see the future to make this affordable for more patients?
So one, I'm going to say it first because it's what I do, is academic non-for-profit manufacturers. You know, the fact that we are non-for-profit means our products are cheaper, simply said. That is one solution. Another solution is using allogeneic products. Another solution could be not using vectors. Not using viruses. Sorry, vectors, you need vectors, but not viruses. Viruses are expensive to manufacture. So we're using other systems like transposomes, electroporation, etc. So there are cheaper ways of transfecting T cells. Other options are reducing culture time, because cost is a measure of how long does the product need to be in the bioreactor. In the future, I hope the technology will improve and we will be able to have better, safer and cheaper bioreactors as well. So again, there are so many components in the CAR T cell manufacturing. There are many ways in which costs can be reduced. If you can think of every single part of the procedure, you'll find ways of improving that part and reducing the cost.
Are there attempts to inject the patient directly with a construct that will integrate in their cells without having to remove the cells from the patient?
Yes. I'm not aware of this being done in patients as of today. Perhaps it is, and I'm unaware. But that is something that several biotech companies are developing, and it could certainly be the future. So you don't need a bioreactor anymore. The body of the patient becomes the bioreactor. And not only that, the cells will be healthier, because the longer they stay outside the body, the worse. So absolutely, this could be a good way of shortening the time and reducing costs as well. But I am not aware of this being done, not even in a clinical trial, in a patient. It's been done in animal models, that's for sure. And there are several companies and academic groups working on that.
Is there anything else you would like to add for the audience about how to streamline this process, or what can we look forward to make this process better?
What I would like to say to the audience is something that I like saying all the time, which is that CAR T cells and ATMPs in general are products that can be manufactured in the academia, and that we, people working in academic centers, may take advantage of that. So there's no doubt that for most drugs we administer to patients, that is pharma companies' role, and we don't have anything to do there. But for ATMPs, most ATMPs have been developed by academic institutions. So I'd like to say to people, it's possible to do it. You just need a regulatory mindset. If you are serious about developing your product, you have to behave as a drug developer. So for instance, if you're serious about setting up a CAR T cell program, go to the regulators first thing. Ask them what preclinical test I need to do. This has nothing to do with a test you would need for a nice publication in a high impact journal. Completely different stuff. So if you're serious about this, do exactly what regulators tell you to do. If you're serious about developing your product, talk to them about your QC, what quality control tests you're going to use. And the same applies to the design of the clinical trial and basically everything you do. So my main message here is academic, non-for-profit, CAR T cell manufacturing, or ATMP manufacturing for that matter, advanced therapy manufacturing is possible, but you need a switch in your mindset. With that switch and a big team and collaboration, you can do it. And I'm certain about that. I'm truly positive about this statement, no doubt about it.
So thank you very much for this very enriching conversation from your perspective. And so thank you very much, Professor Delgado, for sharing your time and your expertise with us. And thank you all for listening.
Thank you very much for having me.